So good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson. I'm the Executive Vice President here at Scripps Research, and it's my pleasure to introduce you to a lecture by our distinguished faculty member, Susana Valente. So Susana is on the faculty at our Scripps, Florida campus, and she's an expert in treating HIV. She is developing new kinds of medications that might be the future of what we can hope to accomplish. So HIV has been around now for many decades, and there's very effective treatments, and you'll hear a lot about the anti-retroviral therapy, combination therapy that's used to treat HIV, but there's no cure, and there's not really yet an effective vaccine. So here at Scripps Research, we do basic research, and because of the intense focus for many decades onto HIV, it's really become the preeminent virus. It's the virus that we know the most about, and yet there's still a lot to learn. So Susana is going to tell us about her new block and lock approach that is used to combat the latency of the virus that still exists in the bodies of infected patients, even though they may not be symptomatic and have had successful treatment. So Susana got her undergraduate degree in Portugal, and then she did her PhD at the University of Paris and did a postdoctoral study at Columbia University in New York. She joined our faculty in 2009 on the Florida campus and was promoted to tenure, and she's now an associate professor. And she's one of a dynamic group of virologists that we have on both campuses studying all kinds of things, including HIV, hepatitis C, and of course, coronavirus. So without further ado, I'm going to introduce Susana and let her give the talk. So looking forward to your lecture. Susana, take it away.
Thank you, Jamie, for the very nice introduction. Hello, everyone. Thank you for joining the Front Row Seminar Series today, and I'm very happy to tell you about our work on silencing the HIV reservoir. So I'm going to start with some basics on HIV and how HIV replicates, so we are all on the same page throughout the seminar. So HIV is a retrovirus, and it actually belongs to a genus called lentivirus. So this literally means that this is a very slow virus because it takes such a long time to produce adverse effects in the body. HIV was estimated to have been introduced in the human population in the late 1940s, early 1950s, through the chimpanzee version of HIV, which is called SIV, which stands for simian immunodeficiency virus. This was the result likely of the African hunters butchering and consuming ape meat at the time. So the virus particles, it's shown here in this schematic, is surrounded by a lipid membrane that he kind of hijacks from the cellular membrane. In this membrane, he has a series of proteins that are called glycoproteins of envelope. And these are very important proteins. They allow the virus to recognize what type of cell it's going to infect. Inside this protective envelope, the virus has a capsid, which contains inside the capsid basically the brains of the operation. The brains of the operation of HIV is viral RNA. It's not a DNA virus, it's an RNA virus. And inside this capsid, there's also a series of enzymes that the virus needs in order to infect the cell right off the bat before it even has time to make more viral proteins. So those are like basically the minimum requirements for this virus to replicate in a cell. So if you look at this picture here on the right, you see that this is an image showing that this virus is covered with these spike proteins, these envelopes, and there's also a lot of sugar around this envelope, which allows the virus to basically hide from the immune system and escape. This virus is also very prone to evolution. It evolved very quickly, so there is a series of variants of these envelopes on the surface of the virus. So the HIV virus likes to infect what we call helper CD4 T cells. So those are cells from the immune system that are very, very important to promote good immunity. Basically, they help other cells to produce antibodies. They help other cells to kill infected cells and so forth, or to call all of the army of cells that is needed to fight an infection. So the first thing the virus does when it encounters one of the cells is that these little viral proteins on the surface of the cell recognize this receptor called CD4. So that's why they're called CD4 cells, because they have this receptor as well as another co-receptor, which can be CCR5 or CXR4. So this virus recognizes we call it the receptor as well as the co-receptor, and this allows the virus to get inside the cell. So once it gets inside the cell, it brings along those enzymes that are important for its for the virus to copy itself from RNA into DNA. And during this process, it kind of goes inside the nucleus. This is the representation of the nucleus of the cell. The nucleus of the cell is like the cockpit, where everything happens, where the DNA is, all of the information of the cell is. And so the virus has this ability to integrate into our host genome, and that's game over. When the virus gets inside the nucleus, it's going to stay with us forever. This is very, very different than what is happening right now with coronavirus, where all of the viral replication happens here in the cytoplasm, and there's not this insertion into our DNA, and that's why it's so hard to eliminate HIV, because it's once it becomes integrated, it's part of us. And it's partly regulated by some viral proteins, but it's also regulated by a lot of the cellular machinery. So once integration happens, the virus then becomes sensitive to cellular stimulus, and so it can then produce more viral copies. These viral RNA copies exit the nucleus, go into the cytoplasm. Once in the cytoplasm, proteins of the virus are made. They start to assemble under the membrane, and then they finally bud. So these viruses that bud then go on through the circulation and infect other tissues and spread very nicely. So each cell produces a large quantity of virions that can go anywhere where there's a CD4 T cell. So I wanted to put here what the course of an untreated infection looks like. So before the advent of antiretroviral therapy, this is what HIV looks like. So basically, in the first few weeks of HIV infection, in this graph we have represented here on the left in blue the number of CD4 cells in the plasma of infected individuals, and in the right side we have viral RNA per ml of plasma. So basically, during the first weeks, there is this very nice sharp increase in viral replication that is associated with this very sharp decline in the number of CD4 T cells. And this is because while the cell is replicating these massive amounts of virus, it dies along the way. So there is this very nice, very sharp decline in CD4 cells. At this point, some individual will experience the HIV acute syndrome, which is nausea and fevers and so forth. Eventually, the immune system fights back, and there's a very nice sharp decrease in viral loads that is accompanied by a small increase in the number of CD4 cells, right? So over the next period, that can last from 10 years to 15 years, we call the period of clinical latency. That's why we say that it's a lentivirus. It's such a slow virus to produce adverse effects. During this period, the virus will go on and replicate very slowly, and the number of CD4 cells will decline very slowly, to the point when we hit a threshold of less than 200 cells per microliter, when the immune system is very, very weak, and the individual then is going to be susceptible to other infections, opportunistic diseases, and the individual is probably going to die from those opportunistic diseases. And that's what we call the AIDS phase. So antiretroviral therapy has changed things dramatically. So it was introduced in 1996, and it was formalized by the Food and Drug Administration in 2001. So antiretroviral therapy consists of the use of several drugs that target different steps of the life cycle. At least 2 of them are present. Normally, there's 3 drugs. The first treatment that is given to patients, the goal is that we keep viral loads to below 50 copies per ml. If this first treatment fails, subsequent treatments are less likely to succeed, because as the virus accumulates mutations and other drugs are likely to not be so efficient. So without any treatment, without antiretroviral therapy, about 9 out of 10 people with HIV are going to progress to AIDS within 10 to 15 years. Some of them progress even sooner than that. Once HIV has progressed to diagnosable AIDS, the average survival time with antiretroviral therapy is estimated to be 10 years. Without therapy, the individual will normally decease within 2 years. So The anti-retroviral therapy that we have right now in the clinic does not target a lot of different steps of the life cycle. So there's some drugs that target the entry of the HIV into the target cell. So those are a little harder to use, these kinds of drugs, because of the way they are delivered. So they're not that currently used in the clinic. Reverse transcriptase inhibitors, they block the transition from RNA to DNA, and they are very widely used and very good. Integrase inhibitors, or the latest types of inhibitors that came into the clinic, they are very good, and they block the access of the HIV to the host genome. And finally, we have protease inhibitors, which what they do is that they prevent the maturation of the viral particle. So basically, the virus has this core that contains all the genome inside, but if it has to be assembled very, very precisely. And if we prevent some of those proteins to assemble properly, to mature, to be degraded in a certain way and assemble properly, the virus is going to be ineffective. So it can produce virus, but those viruses are not infectious in another round. So you will notice that there's this important aspect here between after integration into expression of the proteins in the viral RNA, there's really no drugs that target this step of the life cycle. So once the virus has inserted itself into the host genome, there's really nothing right now in the clinic to prevent expression of viral proteins or viral RNA. So this is a very, very important need in the field. So if we look at the different types of antiretroviral therapy over the years, things have really improved. So if we look at an individual who was diagnosed, for example, when he was 20 years old, back in 95, 96, he was expected to live in approximately 8 years after diagnosis. Over the different versions of antiretroviral therapy that have been improved over the years, now an infected individual of 20 years old can be expected to live another 55 years, which is basically on par with their negative counterparts. So antiretroviral therapy has really, really changed what used to be a fatal disease to transform into more of a chronic disease. Like I said, antiretroviral therapy does an amazing job so if this is the representation of the circulating virus in plasma of a certain individual, if you add antiretroviral therapy, very, very quickly, you see those levels go below a certain threshold. So it does an amazing job in terms of reduction and suppression of the viral RNA. However, during this period, some individuals will still experience blips or moments of detectable viremia, and these blips are probably just reactivation of the cells. Whether they can contribute to reinfection or not is still outstanding. So, but unfortunately, the individual that is on antiretroviral therapy has to stay on antiretroviral therapy for the rest of their lives. And the reason is because as soon as they stop therapy, there's a full-blown viral replication back to pre-ART levels. And the reason for that is that ART can suppress the virus, but cannot really eliminate the virus from the genome, because the virus remains latently infected. And what do I mean by that? So here, during an active infection, the virus is expressing the RNA and proteins. And so there's proteins of the virus on the surface of the cell. And so the immune system knows that this is an infected cell. Now, during latent infection, the virus is dormant, is sleepy, there's nothing being produced. And so there's nothing telling us that this cell is infected. And so it's very hard to devise strategies to go after these cells, because we cannot distinguish an infected cell from an uninfected cell. Even if we wanted to devise specific therapies just to go after these cells, we don't have a marker on the surface that tell us this. So the only way we can have a marker is by waking up the virus, by making it produce virus all over again. So in the presence of antiretroviral therapy, we have a lot of cells in this stage of latent infection. But as soon as antiretroviral therapy is stopped, we move the needle in to become an active infection. So there's several ways that this latent provirus can persist inside the human body. The main way that it persists is basically by what we call homeostatic proliferation. So basically, HIV infects memory CD4 T cells. So those are the cells that are responsible for immunological memory, and they have a very long half-life. So it is they proliferate, they are maintained. Once in a while, when they encounter an antigen, they can amplify, and the virus takes advantage of that to live a very long life inside these cells. Another hypothesis that has been thrown out is that there's an ongoing viral replication, even in the presence of antiretroviral therapy, in certain sites where the concentration of drug is not high enough. So it is this is still debated in the field, and some people believe there's still ongoing viral replication in specific sites that allow the maintenance of these latently infected cells. So the sites of viral persistence are many. Wherever you can find a CD4 cell, you can call it a viral reservoir, because it can trigger de novo infection all over again. But the main sites of persistence is the brain, the lymph nodes, peripheral blood, the gut, the bone marrow, and the genital tract. So we've been putting a lot of effort in terms of devising strategies to cure HIV. And we can think about the cure for HIV in two ways. We can think about an eradication, which is basically what we call a sterilizing cure, where we remove all of the virus from the body. And this is really what we would love to see in our lifetimes, is that we find a sterilizing cure for HIV. So far, though, only 2 people have been cured from HIV. The first person was Timothy Bray Brown. Timothy Brown was diagnosed with HIV in the late 90s while he was living in Berlin. Later on in 2007, he developed leukemia, and at this time, his doctor chose from, he required a hematopoietic stem cell transplantation, and his doctor opted to choose cells from donors that had a mutation on the cell, on those receptors that the virus uses to get in, that did not allow the virus to replicate. So it chose cells from these donors, and it transplanted in Timothy Brown. So Timothy received 3 of those transplantations. He also received a lot of conditioning for the transplantation. He went through a series of procedures that are quite difficult, but eventually he was considered cured from HIV. He was called at some point patient no more. And because he offered himself to a series of clinical trials and studies, and no one could really find HIV in his body anymore. Unfortunately, though, Timothy Brown just passed away in September from complications from leukemia. It was a great loss, because he was a real he was a very important promoter of research for a cure. Later on, in 2017, another patient also went through the same procedure called he was called the latent patient at the time. He went also through the same stem cell transplantation, and he received also cells that did not have the CCR5 receptor. So he's also been deemed cured, but it's only been 3 years so far. So another way we can think about the cure is a functional cure, or more of a remission control. This is what we often hear about for cancer. So in this case, the virus is still inside the genome of an individual, but it does not rebound when we remove antiretroviral therapy. This individual cannot transmit to another individual, there's no transmission from mother to child, and so forth. And there's no side effects of the disease. There's no chronic immune activation or any of the side effects of the disease. And this has been observed in a few individuals, namely the Visconti subjects, as well as the French teenagers. So these were individuals which were infected. They received antiretroviral therapy. And after a certain period of time, they opted to come out of therapy. And since then, they are able to control viral replication. They call post-treatment controllers. In this case, there's been more than 12 years going, and they have not rebounded. So they have what we call a functional cure. Now, if we put things in perspective, we have approximately 37.9 million people living with HIV. We have 2 cases of viral eradication and 100 cases or so of HIV remission. So we still have a long ways into finding a way to really cure HIV. But I think in the near future, probably a functional cure or remission are more feasible in the near future. So nowadays, the way we typically assess the efficacy of the different strategies for a cure is to perform what we call treatment interruption. So whatever the treatment is performed during ART here, and at some point after that procedure is done, we remove antiretroviral therapy, and we look at how long it takes for viral rebound. So here in red, you see that a typical rebound occurs within 1 or 2 months after treatment interruption. Here in blue, you have, for instance, a couple of examples of what they were called the Boston patients. So these were patients that received also stem cell transplantations to treat leukemia, but instead of receiving from donors that had a mutation in the receptor for HIV, they received fully normal with normal receptors, and eventually they rebounded. So there was no control of the virus. Other instances of early treatment, for instance, the Mississippi baby, you might have heard this in the news. It was treated very, very early after birth from an infected mother. He was kept on therapy for a period of time, and then he stopped treatment. And for 28 months, we didn't see any viral rebound. So there was hope at the time, but then it eventually rebounded. So these were examples where early treatment can promote a small size of the reservoir that may be able to help with control. And then finally, we have the Visconti patients, or the French teenagers. So those are individuals that they were infected, they were on treatment. For instance, the French teenager was out of treatment and the viral rebound, and then got back on treatment and for a period of time. And some of them for more than 12 years now are able to control the rebound without drugs. So those are a good example of what we could potentially achieve with drugs, create this type of post-treatment control. So here in purple, we have both the Berlin patient and the London patient. So those are examples that we expect them never see viral rebound in these individuals. Those are eradications of the virus. So one of the approaches that was the most explored in the last maybe 10 years is something called shock and kill. So like I said, the cell that is latently infected is hidden from the immune system. So the immune system cannot see it, cannot get rid of it. And so the idea at the time was that we were going to wake up the virus in the different tissues, in the different reservoirs, with latency reversing agents. So this was molecules that would promote production of viral RNA and proteins. So these cells, they were now producing virus. And then we would have a kill phase, which the idea is that the production of the viral particles will eventually kill the cell, as well as, because they're expressing now proteins of the virus on the surface, the immune system would see them and it would eliminate those infected cells. So unfortunately, this type of approach did not work so well. This is a series of 6 clinical trials that were done testing different latency reversing agents. You don't have to worry too much about the details of the graphs, but just I want you to notice that before the treatment and after the treatment, we have a flat line. So this is not good news. Means that after the induction of reactivation, there was no change in the size of the reservoir. So the individuals had the same amount of latently infected cells before and after. So flat lines are not good here. We wanted to see a decrease. So there's several reasons to explain why this didn't work. The first one is that the levels of reactivation that need to occur in order for the kill to happen, either by cytopathic effects or either by the cell dying by itself or by being killed by the immune system, need to be sufficient, and the reactivation was not sufficient. It's also risky in terms of some drugs, not all, can activate other genes, and this can lead to unwanted reactivation of different genes that should not be on, and this could cause side effects such as cancers. HIV latency is also an heterogeneous process. So sometimes it takes one treatment with a latency reversing agent. Sometimes it takes two. How many shocks does it take? We will never be sure whether we eliminated every single cell or not that is infected. Another issue is that suboptimal ART concentrations. ART stands for Antiretroviral Therapy, and I'm going to use that a lot. ART may lead to reinfections. For instance, if there's a reservoir tissue where the concentrations are not optimal, if we promote reactivation with latency reversing agents, those viruses can go on and infect another new cell and create more reservoir. So that can be also a complication. And then finally, it's risky in some reservoirs, for instance, in the brain. So the HIV infects the brain. The brain is already suffers in some cases HIV neurocognitive disorders due to the expression of viral proteins and so forth. So and there is no immune surveillance in the brain. It's very poor. So the kill part, where the immune system would kill the infected cell, is not working properly in the brain. So this could lead to more problems in the brain. So we need better strategies, different approaches to tackle HIV. So we are interested in strategies that keep the virus in hibernation, latent, instead of reactivation. So what we will be looking more like a functional cure rather than an eradication. So what we've been proposing in the last few years is this block and lock approach for a functional cure. So the idea is that we would take the cells that are latent, but they're still trickling a little bit of viral RNA and can promote any kind of reactivation at any time point, and use drugs called transcriptional inhibitors, as well as work with the epigenetic control of the cell, which I'm going to explain in a little bit, and put basically a lock on HIV, not allow the virus to reactivate in the presence of these LRAs or when the drugs are removed, and the person individual would not be able to transmit to another individual. So basically what we want to do is to push things into this direction, not only to silence the virus, but then put a lock on it and basically prevent the virus from reactivating. So how does this sound like? This is not such an odd idea. So if you think about it, in human cells, the default state of gene expression is off rather than on. So it takes more energy for the cell to make proteins and to transcribe DNA into RNA, make a protein, rather than turning it off. So it costs less energy to the cell. And if you think about it, each individual cell we have is probably has an average of 20,000 genes. And per cell, around 80,000 are expressed. All of the others are silenced, they're not expressed. For instance, only the pancreas can produce insulin, not other tissues. So how does this work? So the secret of this lies into chromatin. So chromatin is a set of proteins that's wrapped, where DNA is wrapped around, and they can be tighter or not. And when they are very tight, they do not allow the machinery of the cell to access the DNA and to copy it and to make proteins. So this closing or openness of the chromatin is called epigenetic control, and can potentially control what is expressed in a cell. So how would this look like in terms of therapy exactly? So right now we know that we have very, very efficient antiretroviral therapy to block the virus. So the idea is that we would add these novel types of inhibitors that don't exist yet in the clinic, called transcriptional inhibitors. This would basically silence all of the RNA that is still being produced in the presence of R to very, very low levels, to achieve a place where it's so low where the chromatin becomes all packaged and does not allow the virus to rebound in the absence of drugs. So this is basically what the block and lock approach would look like. So if I want to explain to you transcription and proteins and so on, my great student Louisa Mori came up with this idea of representing things with a little car. So the idea is that, so we have a gene that needs to be made into a protein. So there's this protein called RNA polymerase. So basically that RNA polymerase is recruited to the gene that needs to be produced. And all of the transcription factors and epigenetic regulators are kind of like the sign, the light switch. So first this car needs to get going in kind of first gear. And especially for HIV, this first transcription of the virus is very weak, but the virus kind of goes through a full round of transcription very hardly. When it does so, it produces the first molecules of RNA. And one of the first proteins that the HIV produces is this protein called TAT. And we can imagine TAT as like fuel to the car. So if the fuel comes back to the car, fuels the car, and boom, the car can go in fifth gear and transcribe a lot of RNA and make a lot of virus and make a lot of TAT. And the protein goes back and feeds into the virus and more into the RNA polymerase, and more virus is produced. So this is a feedback loop, and TAT is really important in amplifying this vicious circle of the virus. So we have two ways that we have been studying on how to block this car to go through the HIV genome. One of them is by inhibiting the viral TAT protein using a small molecule that is called dihydrocortistatin A, and for short we call it DCA. The other way is by using an FDA-approved drug called spironolactone that actually degrades a protein, XPB, and this protein is part of those proteins that are required to bring even the car to start going in first grade. So basically what they do is this. So spironolactone kind of prevents the car from kind of even starting first gear. And DCA, on the other hand, blocks TAT, basically blocks this amplification of the virus. So with DCA, so it is exactly this. So TAT is kind of the fuel, triggers the transition from latent infection to an active infection. And with DCA, we can reverse this back to latency and even push it even further down. So we discovered this drug called cortistatin that can inhibit HIV. Is a very potent inhibitor of HIV. We did not discover the molecule. We discovered that the molecule blocks TAT. So this was initially isolated. Cortistatin is a natural product. It was isolated from a marine sponge called Corticium simplex that can be found in Southeast Asia. And here's the structure of the molecule. It was found, people found interest in this molecule initially because it had anti-tumor activity in certain cell lines. But it was very hard to isolate. It took approximately 1.5 kilogram of the sponge to yield 22 milligrams of cortistatin. So obviously we're not extracting it from the poor sponge. And so synthetic chemists devised ways to synthesize cortistatin in the laboratory. And namely Phil Barron in the Scripps campus in California showed how to synthesize gram quantities of the functional analog. So it's basically very similar to this, it has the same activity. It's just slightly easier to synthesize. And it starts from prednisone and it takes certain chemical steps. So I want to show this data here just to show how different these type of compounds are. So in an acute infection where we infect new cells and we add increasing concentrations of drugs, in this case, we added these 3 different drugs in pink and blue and green that are already used in the clinic and very good. And we compared DCA with those drugs. So you can see that the curves are very different for start. So with these different drugs that are in the clinic, you see there is a very nice inhibition to very maximal levels. And with DCA, you have this very nice inhibition at very low concentrations, but it doesn't get to maximal inhibition. And this is because of the nature of the TAT inhibitor. So before TAT is produced, the CAR on the gene can already go a little bit. So we can only stop the amplification, but we cannot stop that first go at it. However, though, when we look at cells that are already infected, and if you treat them with these drugs that work wonders for new infections, it doesn't work to reduce the viral expression from a cell that is already infected. And think about that. Most of the times people only find out that they are infected after the fact. So they already have cells that are infected. So we need drugs that can treat those cells that are already infected. So it is a very, very different type of mechanism of action. So when we take, for instance, cells and we treat them with a combination of ART and DMSO is just a carrier drug. So let's say we treat them with a combination of 3 drugs and DMSO or 3 drugs and DCA. You see that over time, you see this very nice inhibition so the virus becomes silenced. And when this silence occurs, like I said, the chromatin starts to change and around the area where the CAR needs to go starts to get really, really, let's say, rusty. So the CAR cannot even access to the gene of the HIV. So what we expect is that, okay, let's say if we stop ART, can we jumpstart transcription or not? So that's what we did here. We stopped anti-retroviral therapy and we did not see any viral rebound because now the promoter is all, the HIV is all packaged so densely that the CAR cannot get to it. So now we took these latency reversing agents that we are used for the block and lock approach to reactivate the virus. And we tried to see if we could reactivate the virus that was in this very profound state of latency. And what in this case, we used this combination of 2 drugs. And what you can see is that the cells that have been on ART alone, they can reactivate with PMA and However, the cells that had been on this induced latency by DCA, we could not wake up the virus. So the virus is very strongly inhibited. Okay, so that's great. Let's see if this works in cells from infected individuals. So we took blood from infected individuals and we collected the CD4 T cells from the blood and we increased their numbers. And then we did this treatment where we treat them with a cocktail of antiretroviral therapy or antiretroviral therapy plus DCA. And then we did those studies of treatment interruption that I described before, where we see how much time it takes to viral rebound. So here So they were treated with these drugs for 35 days. And here what we're looking is after treatment interruption, can DCA limit rebound or not? So you see these are cells from 5 different subjects. And you see in purple that the cells that received ART alone, they rebound immediately. And the ones that received DCA are very, very nicely controlled. So 7 days after we removed all the drugs, we came in and said, okay, what if we stimulate the cells now again with more latency reversing agents? Can the cells that were pre-treated with DCA really withstand those events? And sure enough, they did. So there was in some instances some blips, but in most cases, all the viral were very, very limited. Viral rebound was very limited. So this was very encouraging, and we moved on to testing things in the humanized mice, in the BLT mouse, which is a version of humanized mouse. So in here, what we did was just treated ART with DCA for just 2 weeks. And I want to emphasize that for these types of drugs, the longer you treat, the better it gets. So 2 weeks is actually a very short period of time. But in this short period of time, we already had amazing effects. So for instance, in the lymph nodes, we saw a very nice reduction in the cell-associated. So this is not viral particles circulating. We're measuring how much RNA is being produced actually inside the cell. So you see there's a very nice reduction in the lymph node, in the blood, in the brain, and so forth. So this was very encouraging. And so we did another experiment where now we treated for a month and we did the study of viral rebound. So after treating for a month, we stopped therapy. And here in red, you have the mice that receive ART only. And you can see that around day 10, they are already producing virus back again. And the mice that received the DCA, there was a group that rebounded a little later, around day 10, 13. But there was a group that took another extra week to rebound. And if we look at the average of the two. So this is the first time we really saw a delay in viral rebound in the absence of absolutely no drug. This is when we normally we see delays with there's antibodies or if there's some kind of treatment still present. Here was in the absence of treatment. This is just the first time where the cell was controlling how much RNA was being produced. So there is additional benefits to TAT inhibitors that I would just like to highlight very quickly. It's like I said, VARS, HIV infected individuals, like approximately maybe 50% will still experience some form of neurocognitive disorder that can go from simple motor skills to full dementia. And TAT plays a big role in those neurocognitive disorders. So by blocking TAT, we can block a subset of those activities that TAT has in the brain. Also, like I said, during anti-retroviral therapy suppression, the virus is suppressed, but there's this still production of viral RNA and proteins. Even though they cannot infect another cell, there is still production of this HIV material that can cause chronic immune activation. So by really silencing the virus, we can stop all of the trickling and can prevent chronic immune activation. And finally, we estimate that the virus might develop less resistance to these drugs because they are dependent on both viral and cellular components. So in order to evade, it's not just the virus that has to evolve, there's also there has more partners that it needs to interact, and it's harder to evolve resistance in that context. All right, so now I want to move on to spironolactone, which is another of the drugs that we have been working on. And like I said, this one basically targets the initiation of the transcription. Basically, does not allow the car to even get into first gear. So the FDA approved this drug in 1959 and has been around for a long time. It treats high blood pressure, heart failure, strokes, kidney disease, a series of things, including acne and hirsutism and a series of things. It's also used for feminizing hormone therapy. So spironolactone competes with the aldosterone receptor for the mineralocorticoid receptor, and that's how it works for those conditions. An off-target effect of spironolactone, which we call SP for short, is that it causes degradation of this protein called XBB. And XBB is a very important part of the TF2H complex, but don't even worry about what it is. It's just very, very important for triggering the beginning of RNA polymerase to start transcribing HIV. Importantly, though, there's another drug called eplerenone. It's very, very similar to spironolactone. It's even more selective for the mineralocorticoid receptor. However, this drug does not degrade XBB, so it's an excellent control to tell us that the effects we're seeing on HIV are independent of the mineralocorticoid receptor activity. All right, so what happens then when we treat cells that are infected with this drug? So as you can see here, this is kind of the same things that we did with previously with DCA. We treat the cells with the control or epleron, which is that other mineralocorticoid receptor that does not degrade XPB, and you see there's no effect over time. Now we treat with the SP, and you see this very nice reduction in transcription of the HIV, and that is directly associated with the degradation. You can see here in the Western, in this figure here below, this black band represents the protein on a gel, and if you degrade it, you see that it goes away, but with increasing concentrations of spironolactone. So now we took the cells to see whether this inhibition was very strong and you could withstand the reactivation with latency reversing agents. So we stimulate, in this case here, we use prostratin, which is an LRA, and and what we see is that here are, in these two first bars, it's very, very low levels of RNA production in the presence of ART as well as ART plus prolactone. Now you reactivate with prostratin, and you can see that the cells that receive ART alone immediately reactivate. You see here on the third bar, and on the fourth bar is they receive spironolactone, so there's no reactivation. So this drug is also very potent to inhibit the reactivation of the virus. So we wanted to see if this was again true in cells from individuals. So we took blood from individuals and we looked infected individuals, and we stimulated here in this experiment with a cocktail of PMA and amycin. And you see here that when we add spironolactone to this mixture, the cells do not reactivate from latency as they do without the drugs. So this drug also works in cells from infected individuals. So because it is a cellular protein we're talking about, because XPB is a cellular protein, and when you start touching cellular proteins, all kinds of things can happen, bad things can happen. So we wanted to make sure that there was no dramatic effects in transcription or all of the viral, all of the cellular RNA proteins that are expressed in the cell, they were not affected by spironolactone. So what you see here is we sequenced all of the different genes in cells that were treated with spironolactone and DMSO. And I'm not going to go through all of them. Just want to highlight the major difference that you see here in colors between spironolactone and DMSO is here in the HIV category. You see that spironolactone nicely inhibits all of the HIV transcripts. And if you look at the other, all of the other genes of other proteins that are involved in transcription are basically not affected by the degradation of spironolactone. So spironolactone has already been actually described by others that was affecting certain genes more than others. And in fact, HIV is particularly sensitive to XPB degradation, and other genes seem to be fine with it, with whatever levels are enough for maintaining transcription of other genes. All right, so we wanted to ask now whether we could actually replace spironolactone as a single drug when we remove all ART. So we treated the cells with ART with spironolactone. At this point here where the arrow is, we say, okay, let's just give spironolactone alone and remove the cocktail of anti-retroviral therapy. So you see in the control cells in light blue is when we removed the ART. There was not a major viral rebound because these cells are chronically infected. But what we saw is that spironolactone by itself was able to control the virus in this state of deep latency without promoting reactivation. So this is unique because it's never been really shown with any other drug that with a single drug you can maintain this profound state of transcriptional inhibition. So this is quite something. Another experience we did next was, okay, let's remove all the drugs, spironolactone included. We stopped all the drugs here in yellow, and what you see is that there's an immediate viral rebound because as soon as you stop treating with spironolactone, you have the production of the protein, and the protein just jumpstarts transcription all over again. So unfortunately, this was not as we wanted, because for DCA we can remove the drugs and there's no viral rebound. For spironolactone, that's not the case. So it is less effective in that sense, but by itself can be used, and it can now be used in combination with DCA, and that's what we're really now exploring. Another important aspect to consider is whether really in vivo, in models of HIV infection, we could really detect this XPB degradation, and we do. And this is some preliminary studies just showing that treating the mice with prolactin orally, we can have this very nice inhibition of degradation. So we're now studying this now. We'll be studying this in different models of HIV, such as humanized mice or macaque models. All right, so in conclusion, I wanted to just reiterate and repackage everything I've said. So block and lock is a viable approach to an HIV functional cure. Both DCA and spironolactone significantly reduce HIV transcription expression from infected cells. So in cells from people living with HIV, DCA and spironolactone suppress HIV. So this is in the relevant cells, it suppresses HIV. In humanized mice, DCA significantly delayed viral rebound by epigenetic silence provars, and now we're doing more studies in humanized mice and in monkeys really to explore the full potential of DCA. Spironolactone alone controls the viral rebound from latency. This is very important finding. And DCA can also, I didn't show you actually this day, but can revert TAT-mediated inflammation. And I also want to mention that more TAT inhibitors are on the way. So it's really important to have more clinical candidates for TAT inhibitors out there. And there's not a single TAT inhibitor in the clinic. And so we really need to push this type of inhibitors forward because of their amazing potential in the clinic. So some questions remain. This is a very different approach to a cure. So there's a lot of questions that need to be addressed. These are very unique types of drugs. They are not like other drugs that exist already in the clinic. So we really need to understand the full potential of them, because how long we treat results in a different outcome. So if we treat shorter, we might have a rebound soon. If we treat longer, the rebound might be later. So we need to understand exactly how long we need to treat to really allow the cell to start depositing these, what we call epigenetic marks, and start to silence the virus. So we really need to understand a lot of things. So we need to understand how long should the treatment be to totally inhibit any signs of viral RNA. We need to understand what is the relationship between the level of inhibition. Let's say we inhibit 2 logs, 3 logs. What's the relationship between that and the time to rebound? Does that translate in 1 year, 2 years, 3 years, so forth? A very important question is whether this can be permanent. Can we remove all therapy, or should we still keep replace the therapy by one single drug, or can we remove all the drugs? Those are very important questions that need addressing. And of course, if it can become a permanent block, that there is no way the virus can reemerge. Another important aspect is whether there's benefit of adding the drug right from the beginning of treatment, because we know that if we start early and if we start strong, we can reduce the size of the reservoir. That's what we saw with the Mississippi baby. Very short reservoir, had some delay in viral rebound. So if we come in with a different drug with a different mechanism of action, can we actually even shrink further that established reservoir and have a very nice time before rebound. So, and with that, I finish here. I want to big thank to my laboratory. And I want to do a big shout out to Luisa, who helped me put together these beautiful images of cells that you saw. A lot of in black, you have the present members in my lab. Some very important contributions came also from postdocs and students. A big thank to everybody at Scripps that participated in many different aspects of this project over the years. Lydie Trotman has also been a very great, amazing collaborator, as well as Victor Garcia, who has performed all of the different humanized mice studies. A big shout out for Ravi, which has been helping us with DCA and trying to improve the drug and trying to make better drugs for HIV. We are working with David Evans to explore this in monkeys, and a big also shout out to Cecilia, which initiated with us the project on spironolactone, and of course thank you to the NIH for the funding. And I would like to also announce the next seminar series, the upcoming series, entitled Accelerating Innovative Medicines in Times of Change. This is by Pete Schultz, which is our fierce leader, President and CEO of Scripps Research. And with that, I will take any questions. Thank you.
Okay, thanks, Susana. Maybe I'll just start with kind of a general question. Where are the reservoirs? What are the latent cells? And then further drilling down, where does the virus integrate in any given cell? Is it the same place? Is it all over the place? And does that present any special challenges for what you're trying to do?
So I'll start with the last question. Where does the virus integrate? So the virus integrates normally in genes that are actively transcribing. So he's a smart guy and he goes where the action is. So he doesn't have to make too much effort in recruiting all the machinery to places that are silent. So he goes into places that are active. It can be anywhere in the genome. HIV integrates are normally in places, but is not known to cause cancers. So that's a good thing. And there's studies where depending if it's in outskirts of the chromatin or more in the center, there's different types of latency as well, but that is a lot more is still being worked on. So basically that's the answer. The virus integrates a little bit everywhere where there's a transcription occurring, and it takes advantage of that. So the reservoir can be anywhere there's a CD4 T cell. So there's sites where there's more memory CD4 T cells, so they're considered, larger reservoirs and more important reservoirs. But anywhere there's a CD4 T cell, it's a potential site for a reservoir. I'm just talking about CD4 cells, but there's other cells that are considered to be reservoir cells, like macrophages, astrocytes. So those others are still debated whether they really have a big impact in terms of rebound or not. But the main ones are really CD4 T cells.
Yep. Okay, terrific. Here's a question, which is, since you know what the viral sequences are that are integrated, could you actually use CRISPR to? So for the audience that may not know, but CRISPR won the Nobel Prize. It's the latest, greatest, hottest gene editing technology. And so someone is asking, can you use CRISPR for a similar approach to excise the virus?
Yes, that's an excellent question. And we are not my lab in particular, but as an HIV lots of scientists are looking into that. So there's two things that we are trying. Either one is go with CRISPR and trying to really inactivate the virus in itself, in its sequence. The other thing is to get rid of the CCR5 receptor, eliminate the receptor so it doesn't allow the virus to get in. So there's some challenges with these type of approaches. Like I said, anywhere in our bodies where there's a CD4 T cell, it could be a potential site reservoir. So these strategies, sometimes we use virus-like particles to put this machinery of the CRISPR inside and then go and find the cell and do the CRISPR. The cells need to go where these particles that we engineer need to know where they're going. And like I said, the latently infected cells don't have any markers on the surface. So either they go to every single cell and search, or ideally they will go guided, like, by something, but that's not the case. We don't have the guide. Yep. So unfortunately, it's kind of, we need a lot of particles of this to go to every single cell to try to find that CD4 cell and excise. So it's quite including the brain. And so it is quite challenging these days.
Yep. Here's a question from Tom Bamberger. So the DCA was formerly a cancer drug, and is there he's curious about the possible synergistic effect with DCA, such that the pool of CD4 positive cells is depleted with the treatment, and does that help?
So DCA is an anti-angiogenic compound, and so prevents the formation of the vessels that go and feed the tumor. And it does so by DCA inhibits a kinase called CDK8. So we have found that there's other versions of DCA that we've made that cannot inhibit the kinase. So they cannot inhibit the cancer, the angiogenesis, but they can still inhibit the HIV. So the two functions can be separated. Whether it is helpful to have the anti-angiogenesis in terms of therapy against HIV that is taken chronically, probably best to stay away from that. So we have now ways that we can have similar drugs that don't have that effect. Whether it where they would be helpful needs to be tested now in larger models like the SIV infected macaque model in order to answer those questions. So we're not there yet.
Okay, here's another question. This is from Guilherme Zandamela, and he's asking about how are the two drugs that you talked about selective for the HIV, and so they're actually different in that respect. But maybe you could just remind everyone.
So DCA targets specifically a viral protein. And so that's great for anti-retroviral therapy, because if you just stop a viral protein, you don't mess around with any cellular protein, that's a good thing. Their specificity. That's why I mentioned that we can, because like I said, DCA can also touch this other cellular protein called CDK8. If we want to stay away from that, we have versions of DCA that don't touch that. So that's great when we have a molecule that is specific for the virus and doesn't touch the cells. Spironolactone, on the other hand, it degrades the cellular protein. Spironolactone, they were not selected for HIV. They were found spironolactone is a repurposing of an existing drug. We found that he had this activity. We found that he did this for HIV. So now we're trying to explore it in that, but we did not select for it. For DCA, we actually did not select it. It was kind of a serendipitous event, and we found it was an inhibitor of TAT.
Mm-hmm. Great, thanks. So what about resistance? So emergence of resistance is a major issue with the RT inhibitors, the protease inhibitors, and so what's the problems and challenges with potential resistance for either of those two drugs?
Okay, so it's an excellent question. We have done resistance studies in vitro, in tissue culture, like in the laboratories, because this drug is not in the patients yet. We cannot do in vivo resistance. We cannot see if there's any clinical relevant resistance. But in tissue culture, it's very interesting, is that it was very hard to get resistance. But normally with any kind of drug, like a reverse transcriptase inhibitor or something like that, within a month, you have resistance to the drug. If you use concentrations of the drug that don't inhibit the virus, that only just half inhibit the virus, over a short period of time, you start getting resistance. With this drug, with DCA, it took us approximately 12 months. And we obtained two viruses that were resistant to DCA. And interestingly, what they did is that they did not mutate TAT, and they did not mutate the piece of RNA that TAT talks to, which is TAR, but they inhibited a series of other things in the virus that allow for using the cellular transcription better. So basically, they develop a way to have a very strong usage of something called an FKB. So they can basically transcribe in the absence of TAT very, very strongly. So they created this ability to transcribe very, very powerfully in the absence of TAT. But the interesting thing about these viruses is that they kind of commit suicide in a way, because they express so much and they are so cytotoxic that they kill the infected cell. And also, if this was to occur in vivo, they would probably be seen by the immune system, and the immune system would eliminate them, would act like a latency reversing agent.
Right.
So they can evolve, but then it might be not helpful for them in the long run. And so, this is in vitro.
Have people mapped the sites of integration in chromatin for latent cells versus actively cells with active replication? Has anybody done that?
Yes, yes. There's and there is some studies which suggest that the frequency was, you know how it is, also depends on the sample, depends on the isolate, all of these different things, but and the technique. But there is differences, like I said, if they're out in the outskirts of the chromatin, they have, there's differences whether there's more latency in those that are outside versus inside and so forth. But those studies are still very few and need to be done with more samples and more additional approaches.
So do you think that if either of these drugs that you're working with became available in the clinic, would you anticipate that you could reduce the levels of the ART therapy?
Yes, that's the
And
goal.
that's but you'd have to add more drugs, but you'd use less of the other drugs, I suppose.
Well, so that's why I say we still need to really see the clinical utilization of it. But I think that over time, adding a transcriptional inhibitor to the current therapy, we could reduce transcription to such a degree that we could potentially go with no drugs at all. We would have basically we would have a period where we have to combine drugs, but then we will get to a point where it's significantly silenced and we know there won't be viral rebound and people could be off therapy for a certain number of years.
All right, we're really coming up on 2 hours. There's quite a few more questions, and if we can't get to all of them, we'll try to get back to you by email later. But I thought maybe just maybe ask a forward-looking question. So you're involved in basic laboratory research, and you've got infected cells from patients, you've got mice, but what's it going to take? What's the path to the clinic? How long does it take? How much money does it take? This is a great new discovery, but in the end, what's it take to bring this into the clinic? And maybe just talk about that a little.
So spironolactone is already FDA approved. So we are moving forward with that. We are, looking at samples from individuals that are already on spironolactone, and we can propose, small pilot clinical studies just to really assess the effect of spironolactone on HIV infection. And it's very so spironolactone is a different. It's more advanced and it's easy. With DCA, it will take to show that it's not toxic in the long run, that you can synthesize large quantities of DCA, etc. So we are still hampered a little bit by the amount of DCA that can be synthesized. And that has kind of blocked our moving forward quicker because it is a little bit difficult to make and it's a little bit expensive. So that's why we're investing a lot in finding alternatives to DCA. So DCA was kind of our proof of concept molecule,
right.
But it has actually very good pharmacokinetic properties, and it could be a great drug. It just needs to be there needs to be some investment in the scaling up of the drug. Right. And I think that that's why we're also investing a lot in doing high throughput screens and finding alternative molecules. And we have been successful so far. We're very happy. We have a few candidates in the pipeline that not only inhibit TAT, but they actually degrade TAT altogether. So that's really even better because TAT, if we just inhibit a portion of TAT, TAT is so tricky, they can do other things in the cell. If we get rid of it altogether, it's even better. So we are really, putting a lot of effort on those. And I think that, takes time to get something to the clinic, takes time because at some point the government doesn't fund those types of efforts. This needs to come from a pharmaceutical company, and sometimes you cannot find grants to fund that kind of development. You really need to be licensed by a big pharma and go with them.
Okay, well, listen, it was a terrific lecture, and congratulations on a pretty exciting and novel approach to inhibiting HIV, and obviously more to do, but it's pretty exciting. So thank you very much.
Thank you very much.
And for everyone else, we have Pete Schultz, who will be giving the front row next year. So the first talk of the year in January will be given by our president, Pete, on Tuesday, the 19th. And I hope everyone enjoys the holidays in whatever muted form they might take over the coming new year. So thank you all for coming to the front row. Hope you enjoyed it, and we will see you next year. Be well.